drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. Hey everyone, I want to share with you a new AI-powered research tool I've been using to find scientific papers and learn from the literature. It's called Consensus. It is an AI-powered search tool built using large language models trained to help you find and understand the best science faster. Consensus helps you not only find individual papers, but its AI models will help you assess research quality and even synthesize insights across multiple papers for you. You can ask it general questions about any topic you might have, such as what are the known risks and benefits of a certain prescription drug? What do we know about how a certain food or nutrient affects the human body? Anything you can think of. Its Pro Analysis feature will actually provide you with a concise written summary of what the literature says, broken down point by point, highlighting the strength of the evidence across papers and providing you with individual studies ranked by the relevance and including additional information like citation rates, journal quality, and more. You can also save, cite, and share individual studies with a single click. Unlike other AI and search tools like ChatGPT, PubMed, or Google Scholar, Consensus uses over 20 custom large language models that are developed and trained for this specific purpose. The base Consensus product is totally free, but the premium product that you can subscribe to has better features and unlimited queries. And I highly recommend this if you are using outdated tools like PubMed or Google Scholar, or you just do your own research. For a limited time, you can use the code MINDMATTERSPECIAL, all one word, to get a free premium subscription for 1 year. That is a $150 value for free if you use the code MINDMATTERSPECIAL today. So click the link in the episode description or check my support page and give Consensus a try. Use the code MINDMATTERSPECIAL to unlock your free premium subscription today.
So I'm Rose Bagot. I'm an associate professor in the Department of Psychology at McGill University in Montreal, Canada. I'm also an associate member of the Department of Psychiatry and a primary investigator in the Ludmer Center for Neuroinformatics and Mental Health, and a member of the Concordia Center for Studies in Behavioural Neurobiology.
Mm-hmm. And so what does your lab do in terms of, you guys study animals? Do you study humans? Are you doing molecular stuff, system stuff? Where do you kind of fall?
Yeah, so almost everything we do is in mice. And in terms of what we do, I'd say that broadly, we're interested in understanding the neural bases of affect and affective behavior. But as I said, we're doing this in mice. And sort of the approach that we take is starting with robust behavioral paradigms in mice. And then we use this as a foundation to explore the molecular, cellular, and synaptic changes that are induced by things like chronic stress or other kinds of experiences, and to understand how that changes neural circuits to lead to changes in behavior. Mm-hmm. So we've done a lot of work looking at how chronic stress leads to changes in the brain with sort of differential emergence of susceptibility or resilience, and using this as sort of a window into understanding mechanisms of stress-related disorders like depression.
Mm-hmm, mm-hmm. And, starting at a very high level, if we think about just animals generally, a little mouse, a human, whatever, just mammals, birds, like just animals in general, we all experience stressors in life. It's just part of being a living thing. when we think about sort of the natural, healthy, ecologically appropriate mammalian stress response, can you just walk us through sort of the basic stress response as it should work under normal circumstances in a healthy individual? What are some of the hallmarks of that?
Yeah, so I'll answer that directly in a second, but I think the really first thing to say is this idea of like a normal or adaptive stress response. Um, there is no absolute. It's really contextually determined.
Mm-hmm.
And I think that's just sort of generally an important sort of caveat to keep in mind. But in terms of like what is a stress response, at the simplest level, we can talk about the HPA axis, the
Mm-hmm.
hypothalamic-pituitary-adrenal axis. And so when an organism, be it sort of me joining a podcast or a mouse in the lab, encounters some kind of stressor, so something that elicits a stress response, we get activation of the HPA axis. And this leads to release of various hormones that ultimately leads to release of cortisol in humans, corticosterone in rodents.
Okay, so we can think of the quintessential stress response as being a measure of how much this HPA, this hypothalamic pituitary adrenal axis, gets activated. And when we say activated, we basically mean stuff is happening in that part of the brain such that a certain cocktail of hormones get released.
Yeah, in a sense, we can. And so this is what we mean by sort of the classical stress response is this HPA axis activation. But most of what we study in my lab is you could consider it as being sort of upstream of the HPA axis. So
Mm,
we're interested in what's happening in sort of higher brain regions that influence how the HPA axis is being recruited.
yeah. Yeah, so like if I'm out in the wild and a tiger jumps out of me, my
Yeah.
HPA axis will light up. I will have a cortisol
Yeah.
spike and all that. But, I had to see the tiger. It had to go through my retina, get in somewhere
Yeah.
into my nervous system before that stress response got elicited. And
Yeah.
so there's sort of probably multiple things happening in parallel here, from the activation of the HPA axis itself to
Yeah.
the object recognition and the sensory perception and
Yeah.
all that.
Yeah. And so the tiger jumping out at you in the wild is a pretty unambiguous stimulus.
Mhm.
And so then you really want this sort of like very acute activation. And there are sort of other systems that are responding to that as well. But a lot of what we're interested in studying is probably more in what you consider the gray areas. And so how stimuli that could be threatening or maybe aren't so threatening, how that could lead to changes in behavior. So I'd say that in a sense, like the HPA axis is in the background of a lot of what we do, but it hasn't been the focus of anything I've done since back in my PhD, where we really were looking sort of more directly at HPA axis regulation. We did some work that the lab where I did my PhD, we were looking at how just very small variations in experience during the first week of life in
Mhm.
rats, and
Mhm.
the experience was how much they were licked by their mother.
Mhm.
Um, how that leads to changes in specifically how the HPA axis is regulated in response to a stressor. And we can look at sort of the time to the peak hormonal response. And then also a really critical factor in HPA axis function is the shutdown, because
Mm.
you need to have this strong response to a stressor. You need to be able to mount a response. But then when that threat has dissipated, you need to be able to come back to baseline.
Yeah, yeah. And I imagine there's a lot of variability between species and between individuals and that stuff. How quickly it comes on, how quickly
Mhm.
it goes off,
Yeah.
whether or not it comes on in response to something that
Yeah.
someone else might perceive as innocuous, you perceive as a huge stress.
Yeah, because yes, you can also have disorders of HPA axis function where you don't mount an appropriate stress response. So
Mhm.
there's lots of ways that the HPA axis can go awry.
Yeah,
Yeah,
yeah. Before getting into some of that and some of the neural
yeah,
stuff, I wanna cover off on a couple other basic things. So
yeah,
you mentioned the HPA axis, and one of the key outputs of that is cortisol. Cortisol is pretty widely known. Most
hmm
or many people out there have heard of
yeah,
cortisol. We call it the stress hormone or a stress
yeah.
hormone. What exactly makes it a stress hormone in the sense of like what is the cortisol doing? What kind of physiological changes does it instigate?
Yeah. So cortisol in humans or corticosterone in rodents binds two different receptors, glucocorticoid receptors and mineralocorticoid receptors. And these receptors are widely expressed throughout the brain, but also throughout the body. And so what does court do? Many, many, many things. So acutely, it's really important in kind of reorienting the organism towards meeting an acute challenge. So you get changes in metabolism, changes in alertness, but chronically, you get a lot of sort of negative consequences where you have this continued activation and continued stress response. So you can get like metabolic disorders. You can even, in extreme cases, have stunting of growth. You have a shutdown of reproductive functions, and you can have atrophy in various brain regions, but also hypertrophy in other regions. So it has very wide-ranging consequences.
Mm-hmm. But among other things, it sounds like the cortisol, it's having hormonal effects that influence how the animal is mobilizing its energy stores.
Yes,
for example.
for sure. Yeah, that's a core function.
Interesting. Yeah. So instead of, so if it's a growing animal, for example, some fairly large percentage of your energy is going to go towards growth and building new tissues and stuff. But if you're stressed a lot, you're going to mobilize that for other purposes, and then therefore you're going to take away from that type of function.
Yeah, absolutely.
And when we think about this on the neural side, I'll let you sort of navigate us in terms of some of the experiments that you've done in your lab that are relevant. But when an animal is perceiving a stressor, before maybe even the HPA axis turns on, what are some of the major neural pathways that are involved in, if it's threat detection or just the perception of a stressor that's, before that hormonal effects of the HPA axis actually start?
So really. So upstream, we're talking about upstream of the hypothalamus. So the hypothalamus is a brain region that's the sort of, you could think of it as the start of the HPA axis. It's the H in HPA. And the hypothalamus receives inputs from many different brain regions. And in terms of what brain regions are going to be processing the different threats, that would depend a lot on the nature of the stressor. And so you could have sort of direct inputs from the amygdala related to sort of like fairly unambiguous threats. We've done a lot of work looking at, I'd say, sort of less traditional pathways about prefrontal cortical and ventral hippocampal inputs to the nucleus accumbens. And so they're not circuits that are traditionally thought of in the context of stress response, but they're pathways that we found are modified by stress. And then coming back to ask sort of what their function is and what they're doing, we've found that they do seem to be quite important in processing specific aspects of threatening stimuli.
What would be some examples of that? Like what aspects?
Yeah. So what we found, so in our most recent work, we kind of took a very reductionist approach, and we're just looking at which neural circuits process an auditory stimulus that predicts a foot shock. So the threatening stimulus is the foot shock. We can look at just the neural response to the foot shock, and we see that both prefrontal cortical neurons that project to the accumbens and ventral hippocampal neurons that project to the accumbens, both of these show an acute increase in neural activity to the foot shock. There are some really interesting sex differences there, but maybe let's come back to that in a second. But then when we can also look at how does that auditory cue that precedes the foot shock, how is that encoded? And we find that, again, both of these circuits encode that. But when we go a step further and we look at what's the response to a different auditory cue that the animals encounter in the same setting, but that's never associated with foot shock. So we can think of it as a non-threat cue. And we see that these pathways seem to encode the identity of that non-threat stimulus as well, but in these sort of curious sex-specific ways. And another sort of nuance to this work that sort of resists simple explanation is that we see that this neural encoding is happening when animals are just encountering these stimuli. But when we look at how that relates to behavioral responses to threat, and so remembering that there are lots of different ways that animals can respond to threat and can respond in a stressful or threatening situation, we see that neural encoding doesn't seem to have much to do with these sort of canonical threat behaviors like freezing, but it seems to be really important in how animals kind of use that information to guide sort of ongoing reward-seeking behavior.
Okay, so if I'm starting to understand this, basically something is going to end up being stressful, which means it's going to end up activating the HPA axis. But
Mhm.
depending on exactly how directly stressful it is, or whether it's not stressful but it predicts something stressful, like the tone paired with the foot shock, there are sort of multiple paths in the brain, multiple circuits that can eventually get you to the activation of the HPA axis. And there's probably different. I think what you started to say there is there's differences between animals that affect things like um, how easily new stimuli are going to end up affecting that stress circuitry. So, for example, if you pair the tone with the foot shock's obviously a stressor. The tone is paired with it, so the tone sort of ties into things. But then it sounded like you were saying that if you just play like another tone, not that tone, but maybe a similar
Mhm.
tone, for some animals, they say, okay, maybe that one's stressful too, but some animals don't do that. So there's this sort of susceptibility issue that seems like it's coming in here.
Yes, but I think there's kind of a couple of things to unpack here. And the first thing is sort of this comment about how different brain circuits sort of lead to activation of HPA axis. One thing to note is, like, in our recent work, we haven't actually been looking at this question of how HPA axis is activated. We're looking more at how, say, threatening stimuli are encoded.
Mm-hmm.
And then one of the consequences of that encoding, you would imagine, is activation of HPA axis, but we haven't looked at that directly. So just sort
I
of
see.
put that aside for a second. But then I think the more interesting thing that came up from your comments is thinking about how, like, different brain circuits can encode different kinds of stresses. And I think this relates to something that is both interesting and very frustrating about stress as a stress researcher. And that is that uh, it's very, very difficult to sort of settle on, like, a definition of what's stress, what's a stressor. And
Mm-hmm.
that's really the nature of the experience, that stress is highly subjective, and we can define a stressor, something that elicits a stress response, by the sort of outcome, how it changes behavior, how it changes court release. But thinking about sort of how stressors, like the stimuli that elicit stress responses, are processed in the brain, is going to be highly variable, because the things that elicit stress are highly variable. So you can have a stress response, you can experience threat from a tiger jumping out at you in the forest, but you could be sitting in your lounge room and just imagining something that's never happened
Right.
and find that stressful.
Yeah.
And so
Watching a
if you think
movie,
about
whatever. Watching a
watching a
movie.
movie, yeah,
Yeah. Yeah.
or even in the absence of any stimuli, just imagining, oh,
Mm-hmm.
I have to do this exam next week, right?
Yeah. Yeah.
And so just appreciating sort of how variable the things are that can elicit a stress response. And I think it follows that the neural circuits that encode those kind of stimuli will be highly variable.
Right. So, I mean, we all can think of examples in our lives. You know, some people really lean into stress. Some people are
Yeah.
stressed out by seemingly everything. So, in other words, the same stimulus can elicit a stress response in
Yeah.
one person, but not in another, or the magnitude could be different, et cetera.
And
And
then I think also how people react to that stress is enormously different. Like, I think some people really seem to thrive on stress and
mhm,
almost can't get motivated without it. And some people find any amount of stress to be disabling. So
yeah,
there's many levels of individual variability at sort of different parts of the process.
yeah. And so all of that sort of it speaks to this idea that um, the way that stimuli are encoded prior to touching the HPA
Yes.
axis is going to affect whether or not that axis lights up, or lights up a little bit or a
Yes.
lot or whatever.
And then downstream, once the HPA axis is activated, that will have very different consequences for different people.
Mm-hmm, mm-hmm. So thinking about like the activation side here
Yeah.
and some of the circuits you were talking about,
Mm.
um, I'm going to ask a bit of an intentionally naive question. So you mentioned
Okay.
the nucleus accumbens as being
Yeah.
an important part here. Normally, when we talk about the accumbens, a lot of people just associate that with reward learning and addiction. They don't
Yeah.
necessarily associate it with stress. Was it surprising for you that this ended up being a place you were looking at, or why do you think that that's a circuit that's relevant here?
Yeah. So the way that I came to it was through work that I did in my postdoc. And so within the lab where I did my postdoc, there was a history of studying the accumbens in the context of addiction and animal models of. and stress. And so there was already like a lot of papers showing changes to the accumbens with stress. So in that sense, it wasn't so surprising, but I agree with you that generally the accumbens is thought of as this reward center. But one way that we can think about sort of why that might be touched by stress is because when you have this experience of chronic stress, one of the sort of key functions that can be impaired is your ability to perceive and to use information about reward to guide behavior. And so I think that's one way we can understand the role of the accumbens in these stress-related disorders.
Mm-hmm. I mean, naturally, I start to think about, when I think about reward and stress, oftentimes the natural response we have to stress and chronic stress is to engage in reward-related behavior. Some people get stressed and they start eating junk food or whatever.
Yeah.
So there is sort of an intuitive tie-in, I would think.
And actually, this is not my work. This is a published work. I don't remember the group, but there's a really interesting paper showing that just giving mice sucrose is sufficient to dampen the activation of the CRF neurons in the PVN, the paraventricular nucleus of the hypothalamus, to dampen HPA axis activity. So probably not a great long-term strategy, but it does suggest that there's like a physiological drive there that consuming sucrose, and this paper only explored sucrose. You could speculate quite likely sort of other high caloric foods might have a similar
comfort food idea.
Yeah, it is actually dampening HPA axis activation, at least acutely.
So basically you give mice sugar and the cells in the HPA axis that release some of these stress hormones are actually less active, basically.
Yeah. Yes.
Interesting. One of the areas I want to get into is individual variability and susceptibility. I know that there's a lot of work out there on some of this, and I'm only partially familiar, but I think a big tie-in here is going to be that a lot of early life experience affects things like the sensitivity of the HPA axis and how much it responds and how quickly it shuts off, whether or not the mother mouse was constantly licking the pup or not, whether there was many siblings competing for milk access, all of these things probably tie into this. But what can you say about the origins of variation in the susceptibility that individuals have to stress?
Yeah, I think this is a huge question. So I think we can all appreciate that individuals show differences in their susceptibility to stress, but why is that so? And I think there are lots of different factors, particularly in humans. We can't discount genetic factors. There's certainly genetic variation that plays a role in this, but definitely life experience is one of the factors. And so, I mean, there's epidemiological work in humans showing impacts of early life adversity, but in rodent models, so I alluded to this work in my PhD, just this sort of natural variation in the amount of maternal care that rat pups experience in the first week of life leads to quite marked differences in HPA axis function. And so differences in the peak and the return to baseline in core release, also ACTH, another stress hormone in the HPA axis. And that then goes on to produce differences in behavior, so differences in anxiety-like behavior. I found some really sort of unexpected differences in how animals learn, and in particular, how animals learn under stress. That suggested that animals that sort of had the lower levels of maternal care early in life showed this paradoxical increase in learning and hippocampal plasticity under conditions of stress, although at baseline they seem to be impaired.
So is the basic idea there that because they had more stress early in life, their brain is sort of tuned in to learning better under stressful conditions or something like that?
So that's kind of like the explanatory model. There's these ideas about preparedness. I think they fall apart if you push them too far. But the idea is that there's this sort of programming that might happen, that if you sort of are born into a high stress environment, you're going to thrive in a high stress environment. I don't think the epidemiological data holds up so much when you look at that. I think there's also a pretty good argument that like compounding stresses actually lead to worse outcomes. But yeah, that's one interpretation of this idea of why you have sort of stress enhancement of plasticity.
Mm-hmm. And, at least from the rodent work, it sounds like, again, if we're talking about rodents, right, we're not talking about
Yeah.
anything
Yeah.
as dramatic as like the major human stressors we might talk about, like being born in a
Yeah.
war zone or into poverty or something.
Yeah.
Just natural sort of normal variation, like a mother mouse has a litter. There's some number of pups in it. I imagine,
Mhm.
if she's got a
Yeah.
particularly large litter, there's only so much time she can give to each individual mouse. Then there's natural variation in how much licking each one gets, how much
Yeah.
cuddling and milk suckling
Yeah.
each one gets. Those types of things are relevant here. That sort of natural variation in early life experience leads to changes in stress
Yeah.
response.
Yeah. So actually, one of my colleagues in the Netherlands, when I was a grad student, did that experiment. It was absolutely heroic. So not only did she observe the rat sort of maternal behavior, I think we used to do it like 5 times a day for 7 days, something crazy. She did all of that, but she also like labeled all of her pups so that she could track exactly which pup was getting the care at each time.
Mm-hmm.
And sort of the upshot of that study was that, yeah, it seems that even so before she did that study, we just looked at the level of the litter, so that little rat family, if you will. But she took it one step further and looked at within litter variation and found that, yeah, it holds up there. So it does seem to be about the amount and quality of care that each individual pup experienced.
Mm-hmm. And naturally, I mean, one human thing this immediately makes me think
Mhm.
of our birth order effects. Those of
Yeah,
us from larger families know that, even though you all get quote unquote the same treatment, there's only so much time in the day. And if you're the firstborn versus the
yeah,
second or third or fourth born, you're just going to get a different level and quality of attention from your parents. And
yeah.
naturally, that's going to affect your development.
So yeah, as a parent of multiple children, I have to say that I am a believer in benign neglect. So not all care is I think we have to sort of see the nuance in this, that it's not
Yeah.
just that we should give all of our offspring more care. Um, but I think the other thing to keep in mind, which I think is super interesting, is that you can also have sort of a converse where you have uh, children or individuals that have like objectively the same experience and yet have different outcomes. And I think
Yeah.
this speaks to the interplay between like gene and environment interactions,
Mm-hmm.
or perhaps some kind of like latent differences that emerge from like prenatal differences.
Yeah. Yeah.
Yeah.
And are there, it sounded like there are some raw genetic, there's some raw genetic variability that might be relevant here. Is there much genetic variability in things like, I don't know, the genes encoding the cortisol receptor or whatever?
That's a great idea. I haven't looked a lot at this because we're working within inbred mice, and
Mm-hmm.
so we
Mm,
really kind of put genetics aside.
yeah.
Um, yeah,
Yeah. So, I mean, independent of any genetic factors here, the fact that you see variability in inbred mice
yeah,
means that there's lots of other things going on.
for sure. And so, like, where does that variability come from in our
Yeah.
mice? I think that it could be related to, as you say, differences in birth order, differences in utero positioning, for instance,
Yeah.
differences in the amount of care experienced early in life. So it could be essentially just the combination of all of the different experiences. But I think there's also an interesting possibility that there could just be stochastic changes
Mm-hmm.
in brain development
Right. Right. Right.
that kind of start on a path that then becomes kind of canalized by later experience.
Yeah, that's an important concept. I've covered it before with people like Kevin Mitchell, but can you unpack that just a little bit more for people? What exactly do you mean by a stochastic developmental effect as opposed to, say, a true environmental
Yeah,
difference between two individuals?
right. So where we're talking about the effect of an environmental manipulation or some kind of environmental experience, we are thinking about the effect of a specific stimulus that somehow impacts the brain. So it has an effect on the brain, but there is an external stimulus. But with stochastic differences, essentially, largely in the absence of the input of specific stimulus, there's just some kind of differences. So we could just think about, say, the normal developmental processes unfolding, that maybe there's just some kind of variation, perhaps in the timing of that, in how different genes turn on and off, or perhaps in the growth rate of different sort of neuronal projections. So there could be all kinds of different biological processes just that are unfolding, and there's sort of some small variation in that could lead to individual differences.
Mm-hmm.
It's not something that I've researched, but I think it's always important when we're trying to understand a mechanism that we kind of think widely about what it could be that's not what we think it is,
Mm-hmm.
right? Because it's very easy to convince yourself, oh, the evidence supports my idea. But I think we should always be thinking about, but what else could it be? Because we're generally less disciplined about looking for evidence for our unfavored ideas.
Yeah, and I think when it comes to thinking about things like developmental stochasticity, our minds as just animals in the world, they're naturally tuned into looking for cause and effect and agency, and A goes to B. What you're saying is, you've got genetic influences, and we all know what that is. You've got environmental influences. Maybe mom was eating a different diet for me than she was for you, et cetera, et cetera. But also, there's just random differences that crop up based on, a neuron going this way versus that way for sort of purely random mechanistic reasons.
Yeah, exactly.
What about, so another element to the variability in susceptibility to stress. So there's this sort of developmental stochasticity thing. There can be genetic variability between individuals. There can be these sort of birth order and other environmental effects that affect, you versus a sibling. What about the role of sex here? I know that there's quite a lot of differences between males and females across species in terms of at the population level between, the magnitude of the stress they experienced, how susceptible they are to certain things, eliciting a stress response, et cetera, et cetera. What can you start to tell us about male-female differences here?
Yeah, I think that there are profound and, at least until recently, underappreciated differences. And so in our research, we talk about the effect of sex because we're studying mice and they have sex, they don't have gender. But when we're studying humans, we also need to think about gender. And that is this additional level of complexity, thinking about the social construction of gender identity. So I think the simplest thing to say is that we know that there are interactions between sex and acute stress response, but also in susceptibility to chronic stress. And so this is something that we've grappled with a lot in my lab in thinking about how to study both males and females. And so not even explicitly looking at sex differences per se, but just studying both sexes. And what is immediately apparent is that doing the same thing to males and females doesn't lead to the same outcome.
Mm-hmm.
And so this could be, say, if we apply like a stress protocol of, say, something like chronic variable stress, we know from work done by Georgia Hodis and Quincy LaPlante some time ago that females show susceptibility, so they show behavioral change after just 6 days of chronic variable stress. And this is sort of repeated experience of, say, foot shock, tail suspension, and restraint stress.
And when you say chronic variable stress, you mean, so the stress is chronic, meaning it's going to last over a fairly lengthy period of time, but it's variable, so they don't really know when the stress is coming.
Yeah, so the chronic is that it's repeated, and the variable is exactly the stress that happens each day. It differs. So that particular paradigm is a pretty simple one where each day the animals are exposed to a stressor for 1 hour, and the identity of that stressor, that specific manipulation, varies each day and sort of just rotates every 3 days. So after 6 days of this, females already show behavioral change. So they show like increased anxiety-like behavior. They'll avoid the center of an open field. They show reductions in social interaction. But male mice take 28 days to show similar levels of behavioral change.
I see. So they get to the sort of same output, but it takes a much different period of time.
Yeah, exactly. And so then that in itself is interesting, and we can study the mechanisms for that. What we've been doing in my lab is asking questions about, so we had a paper looking at the state of susceptibility in males and females, where we were asking if we can induce kind of similar behavioral levels of susceptibility, do they have similar or different mechanisms? And so there we use different lengths of stress to induce similar levels of behavioral change in male and female mice. And then we looked in the brains to see, with this similar presentation of susceptibility, do we see the same neural pathways being recruited?
And
we do. Yeah. So this is a paper where we found that neural activity in the ventral hippocampus to the nucleus accumbens, so this same pathway that we keep coming back to, we found that exposure to chronic variable stress, so we actually used 4 days in females or 21 in males, increased activity in this pathway to similar levels in males and females, despite this sort of differing amount of stress. And looking into the mechanism of this. So first we found sort of this increase just using fiber photometry, which is this in vivo imaging technique, but using slice electrophysiology to try to understand the synaptic mechanism. We found that it seemed like what was happening with chronic variable stress, be it 4 days or 21 days, was increased in presynaptic input to these neurons in the ventral hippocampus that project to the accumbens. So this would sort of lead to the increased activity. But so what was really striking about this is that, I mean, there's a huge difference between 4 days and 21 days, right?
Yeah.
But we saw the same mechanisms being recruited.
Yeah. So it's as if, very vaguely speaking, it's as if the circuit is, keeping track of the total amount of stress. And if the stress gets big enough or quote unquote too much, it kicks in this way. But, how much is too much really depends on, in this case, if it's a male or a female.
Yeah, I think that's an interesting way to think about it. And it also ties into sort of another observation that we made, which is that with this technique in vivo fiber photometry, we were able to do rail recordings longitudinally. So we actually recorded activity in this neural circuit before animals ever encountered stress, and then across the course of stress and after stress. And we found that there were actually individual differences in the amount of neural activity in this circuit before stress that were predictive of that sort of behavioral presentation of stress susceptibility post-stress, which I think sort of comes back to this idea that you're suggesting that neural circuit might be kind of keeping track of stress. So you could maybe it's some kind of readout of some kind of integrated susceptibility factor. Yeah.
Interesting. And that would vary between the sexes naturally and also vary between individuals because of, all of the myriad things we talked about before.
So another interesting thing about that neural circuit, so A.J. Roberson found around about the same time we published this paper, he actually looked at the excitability of these neurons in male and female mice and found that in female mice, these ventral hippocampus to accumbens projecting neurons are actually more excitable. So they're more easily activated. And that's something that we've since replicated in our own lab. So we have individual variability within sex, but we also have this considerable variability between sexes.
And do we have any sense of where this arises? So, one thing that starts to come to mind here. Full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomes, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.